the cell type I'm going to talk about is a lot more differentiated than the cells that you've heard about so far. I think they're still in the library, but they are a mesenchymal stem stromal cell, and particularly focus a little bit on the uterus, but some people don't even believe they are a stem cell, and there's good reason why they think that. A lot of it's to do really with what's being characterised. So let's just define what a mesenchymal stem cell is, or some people call them a stromal cell. The International Society for Cellular Therapy defined them quite some years ago, about 2006, as clonogenic, plastic adherent, multi-potent cells that differentiate into multiple mesodermal lineages, as shown up here. And they express a particular surface phenotype. And that's the unfortunate part, because people hang on to that surface phenotype, and that surface phenotype is on fibroblasts as well as many other cell types. So many people are not working with mesenchymal stem cells, or MSC. Now, why are MSC important? They're important because there are many clinical trials currently in place using MSC for cell-based therapies. In fact, there's over 500 that are listed on the NIH website, and over 2,000 people have had these cells in one form or another for a whole raft of disorders. They're mostly used for disorders associated with chronic inflammation, and they actually rely on the non-stem cell property of these cells, their anti-inflammatory and immunomodulatory effects. And in fact, those components are really often due to the fibroblasts that are in those cell populations that are being given to the patients. So where are MSC sourced from? Originally they were sourced from bone marrow. More recently, liposuction and sourcing them from adipose tissue has become really popular. Both of these are highly invasive techniques, and they require an anaesthetic. And in fact, someone in Australia has died from the liposuction technique. So it's not, entirely safe, even if we've got a lot of fat to get rid of. Reproductive tissues actually offer another source of cells, and these are easily obtainable because some of the tissues, like umbilical cord or the placenta, are waste tissue, and these can be salvaged to obtain these MSC. We focused on the endometrium, the lining of the uterus, of a highly regenerative tissue. These too are very easy to obtain in an office-based procedure, just using a papille biopsy like this. And we discovered in human endometrium, but also in mouse, but in human we discovered the MSC using cell cloning as a technique, just a very basic, simple way to find them. We then went on to characterise these cells for, as it were, adult stem cell properties. And we found that they underwent self-renewal, they're highly proliferative, could produce large numbers of cells from a single cell, and they underwent multi-lineage differentiation. And these 4 lineages that I show you here, they were from a single cell derived, okay? So they really are multipotent cells. They expressed that MSC phenotype, which really doesn't mean terribly much, but and we made the conclusion in the end that human endometrium contained a fairly rare population of cells with MSC type properties. But having clonogenic cells to study is not terribly useful, and we really spent some time to try and identify markers for these cells so that we could actually study them properly and perhaps exploit them for clinical use. Our first set was 2 markers, co-expression of CD146 and PDGF receptor beta, and this isolated a small population of cells. They happened to be perivascular cells, or pericytes, which is the value of identifying markers. And we then went on to screen for a number of perivascular markers to find a single marker called SUSD2, so that we could pull out these cells. This just shows where we could identify the cells as a pericyte here. The SUSD2 is more of a perivascular cell, and we could find them also in postmenopausal endometrium, and they don't express the estrogen receptor alpha. The important thing was the location of these cells in the endometrium. They were around the blood vessels in the basalis layer, which is not shed at menstruation, where we hypothesised they would be. But they were also up here in the functional layer, and this is what's shed during menstruation, but it's also available when you take an endometrial biopsy, in your office-based procedure. So it's a much... we knew then we could obtain these cells very readily for use, potential use in cell-based therapies. And we did all the proper, the tests, the ISCT tests for their MSC properties, but we also were able to reconstitute stromal tissue in vivo. Now, I just like to go through some of the impediments and some of the issues that have arisen, because really what happened with the MSC field is things jumped into the clinic far ahead of the science, and how all the issues are now being raised because they haven't really been as efficacious as one had hoped they might have been based on the kind of properties that they have in vitro. And most of the problem is that they're lacking quite a bit of efficacy in the clinical trials. Much of this is due to this heterogeneity of cellular product. They're really not purified to begin with, and often it's just fibroblasts. And this just shows you, say, fat off that liposuction. It's sort of, as it were, kind of blended a bit and then spun down and given back to the patient. What's given back is just the cell pellet, and it's uncharacterised, and there's a large number of different types of cells in there, besides the perivascular MSC. The other thing that happens if you do isolate the perivascular cell that's clonogenic and has the high proliferative potential is that if you grow them up in culture, which you need to do to get the cell numbers, they'll spontaneously differentiate. And so then you're giving back, again, a small lot of MSC with a large number of fibroblasts. And there's been a very nice study done by Linda Gadish, who's done just a microarray profiling to show that this spontaneous differentiation from the fresh to the cultured cells using these a set of signature genes for the MSC and then for the fibroblasts, and she showed that very quickly in culture the MSC based on our markers, the CD146 PDGF receptor beta population, became like the fibroblasts quite quickly, the CD146 negative PDGF receptor beta positive cells. And the other reason I think why these cells haven't perhaps been as useful clinically is because they're given by intravenous injection. And the thing is, if you give a lot of cells, there's millions, say 70 million cells, if you give them by intravenous injection, they're going to just get trapped in the lungs. It's an incredible waste of dose. 90% are trapped in the lungs. It's all right if that's what you're treating. And they are attracted to the site of injury, but they really disappear well within 48 hours. They do have effects. They have a systemic effect. I think there's a lot of cytokines produced, but all the same, it seems an incredible waste of dose. Then there's the concept of which way do you go, and the business model is allogeneic is really preferable. I happen to favour autologous, which is not a very good business model at all, but if you want to give repeated doses, and even though these cells are lowly immunogenic, they still are immunogenic. An autologous approach, to me, seems a much better way to go. And really, the mechanism of action is not really that well known because, again, the work jumped into the clinic far too quickly ahead of the science. So we're working towards clinical translation of our endometrial MSC, and we're looking at an autologous approach. And we're trying to treat a disorder, which I'm not going to talk about at all today, but it's incredibly common but hidden. It's a women's health issue, and it's called pelvic organ prolapse. And what we're trying to do is culture up our cells. And we've got now our single marker and a very simple way to isolate the cells with minimal damage. We've been working towards culture expanding these cells using good manufacturing practice guidelines. And here we now have installed and commissioned, it's going to be commissioned next week at our institute, the Hudson, a biospheric cell isolator unit. And so this is in place of a big, bulky and really hard to access clean room where you can move around the outside here and grow your cells, isolate and grow your cells in whatever condition you like. And where we've got to with our cells so far, where you have to get rid of all the animal products, we're down to serum free. We're not got rid of all the animal products yet. We've got the serum gone. we find that they grow much better in 5% oxygen and on the fibronectin, and we can generate large numbers of cells quite easily. But the thing is, what we're trying to do now to deal with the problem of the spontaneous differentiation is to block that during the culture expansion process. And we were looking at small molecules to inhibit that differentiation process. And we tried a bit of a candidate approach, and actually we borrowed a lot from the embryonic stem cell literature. And we've been using A8301, and I noticed someone else had that. It was Nathan in one of his culture systems, may have been Mark. But it's a TGF-beta receptor inhibitor, and if you're going to use autologous, you really have to have a standard for your product that's the same and really difficult for every different patient. But this is what we were trying to do, and what we found with the A8301 is it did promote proliferation. You can sort of see this here, just in these microscope views. It blocks, the main effect it has is it blocks apoptosis and senescence, actually. We do get many more clonogenic cells as a result of that. And we find we can monitor our cultures now using this SUSD2. And what we've found here is that when we've treated our cells with the A8301, and these are late-term cultures, and the effect is greater in the older MSC from older women, is that we can bring them up to around about 90 to 95% all the time. So it's for most patients. So this is really quite useful from a quality assurance point of view. We then wondered whether this molecule would be really quite good for all the other MSC types, and so we've been investigating that, and you can see this is very much a work in progress, and we haven't got very many N numbers for some of the ones we've tried. But our hatched bars are where we've treated with A8301, and you can see in all the endometrial ones we get really quite high, and in the placental ones it's improved. And in the bone marrow, which are really quite difficult cells to grow, we make a big difference, but that is only N equals 1 when we're working on it, and we're currently working on the fat as well. Now, one of the other things that we regard as really important before we go into the clinic is to test all these things in vivo. And so Shanti Gurung, who's been doing this work, a PhD student, so just looking at our A8301 treated cells, will they be good and will they be okay after that treatment when we put them into the animal? And so she's just labelled them up with a M-cherry using a lentivirus to track them, put them under the kidney capsule, and this is about 14 days later. You can still see a number of cells there, although it is diminished. And she's been checking with ALU sequences as well, and K's kidney and S's skin, and they're pretty okay. They're at least as much, if not more, than the control cells. So it's still really a work in progress. But the other areas where we're trying animal models is before we go into the clinic with our pelvic organ prolapse surgery, using a tissue engineering sort of approach where we put our EMSC on a mesh and gelatin composite to implant into women. We've been testing them first in a small animal model, and here we've done a fascial wound defect. This is a xenogeneic model, so human cells into rat. Now the cells lasted about 14 days, a little bit longer than an intravenous because it's local delivery, but still not that long. But they did have quite a dramatic effect. They stimulated an early neovascularisation. They promoted quite a strong inflammatory response initially, but that changed to the M1 inflammatory cells to an M2 wound healing type after about 30 days, even though the cells were already gone. And overall, the biocompatibility was markedly increased, the much lower chronic inflammatory response. This is at 90 days. But I think one of the main things that really happened that was really different was the kind of collagen that was laid down. You can see this crimp here versus the scarring type collagen without the cells. This crimp gives the biomechanical properties that are really needed. And so what we found here, and this is some biomechanical kind of testing, but really what you want to see is less stiffness, and this is the slope. So the slope is lower, and a longer period here, and that's kind of the distensibility, and this happens because you've got crimp. and it can be extended. So we were quite surprised at this effect, which is actually only a paracrine effect because the cells haven't really survived that long. But before we go into women, this is vaginal repair surgery from prolapse, birth-induced injury, we want to test our model truly as we will do in women. So we've gone to the ovine model. Sheep vagina is very similar to the women. Here now we're taking the sheep endometrial MSC, putting them on the mesh and implanting them vaginally using urogynecologists to do the surgery. But of course, you know, our SUST2 didn't work. Isn't on, or it's not expressed, or the antibody doesn't react with it. But we have used another bone marrow MSC marker, CD271, to get out some perivascular cells. The perivascular cells are in a slightly different position. They're in the adventitia of these vessels. And this is known in other systems that the adventitial cells, perivascular cells, are also just have the MSC type properties. We've done, this is a very small number, but very encouraging experiment where we put the ovine EMSC, cultured them up, put them on the mesh, inserted it vaginally. This is 21 days later. We had to harvest at that point because this had to go into a grant. But you can see, and you can see we labelled them with an antivirus at this point, and you can see large numbers of cells in a sheet, all looking very viable. So we're feeling that this is really quite encouraging. So this is using autologous cells and local delivery. So just to summarise, in the human endometrium, we have a source of cells from a reproductive tissue that it really is minimally invasive to obtain. It's a highly regenerative tissue, and we think that the cells that we've been isolating are actually involved in the vascular stromal regeneration that occurs every month in these in women. We are developing simple protocols to isolate the cells, and certainly the human cells are much easier to isolate than the sheep with the SUST2. And we're addressing the issue, some of the issues that are facing the MSC field in that spontaneous differentiation in culture by using this A8301. And we're looking really at other molecules too in more of a screening approach. But what that really does is to produce a reproducible product for autologous use, which is quite important when you're having to get something similar or the same from every single patient. And we're trying to determine the mechanism of action of these cells in preclinical models, and we've shown in a xenogeneic model that they have a paracrine action. But in a autologous model, and modelling exactly what we want to do in women, there is the potential, it's still too early to say, that maybe there is some cellular integration because we've addressed all the kind of issues that need to be addressed. And finally, I'd like to acknowledge many of the people in my team, both past and present, who've contributed to the work I described to you today. And in particular, I would like to acknowledge our clinicians, who Anna and Nathania have shown here, who have helped us hugely along the way with advice and with the surgeries. This is James and Shanti, whose work I described quite a bit. But I've done a lot of work with CSIRO, who've designed all the material side of it for us, and they're tissue engineers as well, and also all the funding bodies. So thank you.